I want to thank the organizers for giving me the chance to talk to you today. I also want to apologize to Dr. Toth for unknowingly running away from him constantly, it seems. So I'm going to be taking you all back a step from tafazin and looking at activation of fatty acids in the heart. So the enzyme I study is an acyl-CoA synthetase, which activates fatty acids by the addition of a CoA group. So ACSL1 is the major isoform in the heart and skeletal muscle, and it can activate fatty acids for either oxidation or incorporation into phospholipids or triglycerol. So I'm using a knockout mouse model. It's a tamoxifen-inducible mouse, or knockout, and we induce knockout when the mice are actually adults between 6 and 8 weeks of age. And in the heart as well as the skeletal muscle, we have a 90% loss of ACS activity, indicating that ACSL1 is by and large the major isoform there. In both of those tissues, we see a greater than 90% loss in fatty acid oxidation, but the hearts are able to compensate by increasing their oxidation of glucose as well as changing amino acid metabolism. And we don't see an accumulation of triglycerol, presumably due to an inhibition of fatty acid uptake and retention in the cells. So the hearts are enlarged due to mTOR activation, but they have normal systolic function with a minor diastolic dysfunction. However, the mice live out past 2 years, so they have a normal lifespan. Serendipitously, we did electron microscopy on the hearts to look initially at size and number of the mitochondria, but I was immediately struck by some of the structural changes we were seeing in the mitochondria, specifically that the inner mitochondrial membranes are distorted, as well as we have these vacuolated areas. And so this is a pretty moderate picture, and I was talking to people on my poster last night about some that kind of look like they're exploding, and so I had to show some of those today. So we have some pretty extreme structural defects in the hearts of these mitochondria, or the hearts of these mice. Of course, the next step was to look at function of the mitochondria. So working with Ethan Anderson, I was able to actually look at mitochondrial function in permeabilized cardiac fibers. So this leaves the fibers mostly intact, so we don't lose any of the mitochondria that look like they're exploding, that have the abnormal structure. They're all contained within the fiber yet. So looking at their ability to respond to ADP, it's highly impaired at both of the concentrations of ADP we used, as well as their efficiency to produce ATP is also incredibly impaired in the knockouts. We also measured the ability to take up calcium before the mitochondrial permeability transition, and that is also impaired, indicating a potential susceptibility to apoptosis of these cells. So being in a lipid lab, we immediately started thinking about the membranes of the mitochondria and if there was a problem there. And my advisor immediately started thinking about cardiolipin remodeling. So what we've been talking a lot about is tafazin, of course, here. However, we do know that there's another remodeling pathway which directly uses acyl-CoAs, which got the attention of my lab because our enzyme produces acyl-CoAs. And we knew that when we looked at the ACS activity, so the ability of the enzyme to take the fatty acid and form an acyl-CoA, that the enzyme highly prefers to use linoleic acid, or 18-2. It's actually an eightfold increase over any of the other fatty acids that we used here, indicating that ACSL1 has a huge preference for activating linoleic acid, which of course is of interest in the mammalian heart because we have that high amount of tetralinoleoyl cardiolipin normally. So we looked at the phospholipid species. I'm just going to show you the cardiolipin today, but you can see that the tetralinoleoyl cardiolipin is highly decreased in the knockout mouse hearts. It's about an 80% decrease in the amount of tetralinoleoyl cardiolipin. But if you remember, there's no systolic dysfunction in these hearts. So this was of high interest to us. But we don't see a change in the linoleic acid and the other phospholipids. So we were wondering if ACSL1 is preferentially activating the linoleic acid for incorporation into the cardiolipin. So what I did was an experiment where I overexpressed ACSL1 in this cardiomyocyte cell line, the H9C2s, and then incubated with a radiolabeled fatty acid for 6 hours, and then just extracted the lipids and separated them by TLC. So a very basic method, really, with great results, actually. So after separating the cardiolipin, I was able to see that we don't have a change with overexpression of ACSL1 with either palmitate or oleate, but we have a 20% increase in the label incorporation into cardiolipin with ACSL1 overexpression. So indicating that more 18.2 is being activated and incorporated into cardiolipin. However, we did have also increases in all the other species, especially in PC, which we know can be a donor of linoleic acid for cardiolipin. So the next thing that I did was a pulse chase experiment where I incubated those same cells that were overexpressing ACSL1 with the radiolabeled linoleic acid for 2 hours, and then replaced that with an unlabeled 18.2 for 4 hours. So you can see that with that 2-hour pulse, there's no difference in the amount of 18.2 being incorporated into cardiolipin. However, with the chase, that 4-hour chase, we see that there is about 10% lower label being incorporated into cardiolipin with the ACSL1 overexpression, indicating that more unlabeled 18.2 is being incorporated at that point. So it's the new 18.2. So there's a couple interpretations of that. My favorite being that it's going through the monolysocardiolipin acyl transferase pathway to incorporate the 18.2 CoA directly into the cardiolipin. So to conclude, we found that loss of ACSL1 in the heart induces abnormalities in the mitochondrial structure and function, that we have major changes to the membrane acyl composition, but no changes to the total amounts of either cardiolipin or any of the other phospholipids. We also don't have an increase in the monolysocardiolipin. But we find that less linoleic acid is integrated into cardiolipin in the membranes, as well as ACSL1 activation of 18.2 is permissive for the incorporation of the linoleic acid. But we do see that loss of tetralinoleoyl cardiolipin, that very important species, we thought was very important, is not sufficient for heart failure, but it may be important for the mitochondrial function. And so with that, I would like to thank all the members of my lab, especially Shannon Mentok for doing some of the ACS assays, as well as Ethan Anderson and his student Lalage Katunga for looking at the mitochondrial function and in the heart fibers, as well as Robert Murphy and Sarah Martin at University of Colorado for doing a lot of the phospholipid analysis. So with that, I'll take any questions.
You should write all your talks at the last minute. That's very good.
It's a very interesting talk. I was wondering whether or not you might be able to stress your mice on a treadmill and then find out whether or not you're going to then induce something. Because that seems to me that might be the stress-related phenomenon in terms of the cardiomyopathy.
So these mice also have a loss of ACSL1 in the skeletal muscle. So they're not able to activate fatty acids for oxidation there. So they have an inability to run for long periods of time. When I've stressed our heart-specific knockout, which I haven't actually shown the mitochondrial dysfunction there yet, but they're able to run normally. Their heart function is normal after stress tests. So the heart function seems to be normal with exercise stress, as well as we've done the transverse aortic constriction, so banding of the hearts, and they're also able to they have normal heart function with the banding as well.
Great, good talk. In the pulse chase studies, did you look at labeling that was in phosphatidylcholine?
Yes, yes. And so that does not change with this. We don't see a difference between the control and the ACSL1 overexpression with these time points. Perhaps if we went out to a longer chase, we might see some more differences, but the reason I chose the 2-hour pulse with the radio label is because I didn't want to have a difference in the amount of PC that was labeled to start with.
Hi, I had one specific question, which was just how much reduction did you see in the cardiolipin?
In the total amount?
Yeah.
We didn't see any change in the total
Okay,
amount
and
of
then
cardiolipin.
the
The tetralinoleoyl is decreased by about 80%, but it's the amount of cardiolipin is made up for in other acyl chains.
Okay, and then I had a more general question, which
Yeah.
is you mentioned that the hypertrophy of these mice was due to activation of mTOR.
Yeah.
And as you know, this hypertrophy in mitochondrial disorder is quite common. So I'm just wondering what the data is behind that statement, and if you've tried to block it with rapamycin.
We
Thanks.
have blocked it with rapamycin. We actually just had a paper come out with our heart-specific knockout mice, blocking mTOR activation with rapamycin, and we haven't done conclusive studies with looking at mitochondrial function with the rapamycin yet, but that's in progress.
Do you know if this ACSL is site-specific, like SN1 versus SN2 of my cardiolipin?
I don't know. I mean, I would think that would be due to the downstream enzyme, because ACSL1 is just activating the fatty acid. So whatever enzyme it's then handing the acyl-CoA off to should be determining if it's SN1 or SN2.
Yeah, other question is, do you know if this enzyme is formed with this enzyme in brain, and if you can knock down the same way you do?
So it does not have high expression in the brain. When I looked at enzyme activity, we could see a very small decrease just in the cerebellum, but no other area of the brain. So I don't think it's largely important in the brain.
Stunned silence. Thank you for answering all those questions. So I think for me, this session has just emphasized the amazing strength in depth, particularly with young scientists coming through and doing exciting work, and also the variety of directions that people are coming from. And I'm sure that it really excites Matt and from the scientific point of view. Could I thank you all for all the questions that you've asked and for your involvement in this session.